Welcome to Brain Ponderings, your window into the world of brain research with interviews with guests at the forefront of this field and related fields. And today it's my great pleasure to welcome Professor Dudley Lamming. He's Associate Professor of Endocrinology, Diabetes, and Metabolism in the Department of Medicine at the University of Wisconsin, Madison. He is going to be a great example, this podcast, of someone who's started out in basic research, actually in yeast, and has now moved to work that is establishing what might be the ideal amount and amino acid composition of proteins that we consume in our diets to promote health span and counteract diseases of aging. So welcome, Dudley.
Thanks for having me, Mark.
Okay, so you spent a lot of time in Boston. Did you grow up in that area?
No, I'm originally from New York City.
Ah, okay. And then I see you went as undergraduate, you went to MIT and majoring in nuclear engineering originally, is that right?
And that's correct.
And with also a lot of chemistry courses, I guess. So then you, okay, so that's Boston. So then you did your PhD work with David Sinclair at Harvard, starting out working on yeast. Can you kind of briefly summarize what you were looking at there and what you found?
Sure. I was primarily working on sirtuins. At the time, those were sort of thought to be key longevity regulators. And then transitioned over to starting to look at TOR signaling, which is something that I've continued to work on in mammalian systems ever since.
And was the mTOR, did you start that in Sinclair's lab or was that with the Whitehead with Sabatini and what Bauer?
That was when I started working on TOR in yeast and then moved over to
Ah,
mTOR in mammalian cells when I moved to the Whitehead.
okay, so these podcasts, it's a mix of, scientists at very levels from undergraduate on up, and there's some lay people. Can you just kind of briefly summarize first what sirtuins are? Because there may be a few people that don't know and why they're important in regards to metabolism and aging.
Sure. So sirtuins are histone deacetylases. And basically that means they modify some of the proteins that bind to DNA and help control how different genes are expressed. And they happen to be dependent on a cofactor NAD, which is a key nutrient. And so a lot of research in the early 2000s was focused on the idea that different dietary interventions, particularly calorie restriction, which could extend the lifespan in many diverse species, might work in part by manipulating either the total levels of NAD or the ratio of NAD to various NAD metabolites. And so that was something that I was working on. There's five different sirtuins in yeast, seven in mammals. And in particular, I was looking at some of the ones that are not typically looked at yeast, which were called the HSTs. And so with various collaborators, we showed that these seem to be important in some of their responses to calorie restriction.
Yeah, and this has led, and a lot of others involved, Guarante initially, but led to things like nicotinamide riboside as a potential dietary supplement or the whole resveratrol story or and then Sinclair actually making these trying to get chemicals that can activate these sirtuins because they seem to do good things and counteract
I
aging.
think a lot of people are still working on various different types of sirtuin activators, including various, as you mentioned, supplements of NAD or nicotinamide riboside or other similar compounds that might be able to boost NAD levels.
And one thing we've been interested in, you are too, right, is this idea of, well, one word to talk about is hormesis, but it's essentially adaptive responses of cells to stress, where you subject a cell to stress at kind of a low level, and then the cell activates gene expression pathways, maybe other mechanisms that make them more resistant to subsequent stressors. And caloric restriction fasting is a good example. Exercise is a good example. Um, is there a role, and there's role for these sirtuins in adaptive stress responses, and yeah, there are roles. But do you think some of these like naturally occurring chemicals like resveratrol, we know it's true with like sulforaphane, they typically have a bitter taste. Like if you put the pure chemical on your tongue, they're very bitter. So these are present in like resveratrol, the skin of the grapes, for example, or sulforaphane and like the bitter tasting broccoli sprouts or whatever. And so we evolved with these. So maybe, our cells are geared, they're actually toxins in a sense that they have potential to cause harm, but we've evolved systems to respond adaptively, and maybe the sirtuins is one pathway.
I mean, that may be one possibility. Another possibility is that they might mimic endogenous ligands that we don't know about.
Yeah. Okay, then mTOR. What is it and why is it so important?
So mTOR is a protein kinase, and protein kinase is something that takes ATP and uses it to add a phospho group to various other substrates, most often proteins in the case of mTOR, but there are other different types of kinase that do things like phosphorylate lipids. It's a particularly interesting protein kinase because it acts as a central nutrient sensor, um, which acts to coordinate the availability of different things in the environment with growth and anabolism, or conversely, sort of recycling programs and catabolism of existing things going on in the cell. And so some of those things that mTOR senses includes things like most notably amino acids, but also glucose, cholesterol, various types of things like oxygen and ATP levels in the cell. And it also senses some hormonal cues, most notably including insulin and IGF-1, but some other hormones as well. And if all of those substrates are available for growth and all the hormonal cues are permissive for growth, then mTOR upregulates these anabolic programs that help cells either grow bigger or cells to divide. And so when you think about what a cell needs to divide, a cell needs a lot of protein, and it also needs nucleotides and lipids to make DNA and cell membranes and organelle membranes, respectively. And so some of the things that mTOR does is upregulate ribosomal synthesis, essentially factories for making more proteins. It upregulates translation of different types of proteins as well. It creates different nucleotides and upregulates those synthesis programs, and also activates lipogenic programs as well. And conversely, if some of those environmental cues or substrates are missing, mTOR, when it turns off, will upregulate programs like autophagy that helps to recycle existing proteins and potentially repair damaged proteins as well.
Now, can you talk about why it may not be good to have mTOR like very active continuously 24/7, all the time?
Sure. So mTOR is designed to be turned on and off. When you think about how a human goes through the day, the typical person might eat 3 meals and then sleep overnight. And so every time that they eat, their mTOR activity is going to spike, and then it's going to go down. You could conceive that if you were going to have mTOR signaling on all the time, that would be analogous to a situation in which you're eating all the time, probably not going to necessarily be very healthy. When we think about mTOR being on all the time, that occurs in people with a couple different types of genetic disorders. Probably the most familiar to people is tuberous sclerosis complex, but even so, this is a rare disorder involving mutations that hyperactivate mTOR signaling. And these people have developmental defects, also epilepsy, as well as various aberrant growths in places that there shouldn't be growths, like the lung and the brain. And these can damage the rest of your body by simply taking up available space. mTOR signaling is often hyperactivated in cancers as well. That can happen through a variety of different mechanisms. And so, growth all the time is not good. Um, in the case of people who are particularly old and undergoing sarcopenia, which is age-related muscle loss, we think actually mTOR is actually hyperactivated in that state as well. That we don't truly understand, I think, the mechanisms behind that, but it seems like mTOR gets stuck in the on state, and that continual being on doesn't help muscle repair and respond to exercise correctly.
Yeah, that's really interesting from the standpoint of recommendations for older people in terms of their protein intake. And we can get to that. I mean, you've been working a lot on this. So can you talk about some of your specific experiments, the ones that you think are most important from the standpoint of mTOR, its activation by, I guess, the amount of total protein you intake, but also specific amino acids. So you've been doing a lot of work, which is really important, manipulating specific amino acids in mice and then looking at, uh, well, aging, but also looking, and we'll get to the brain probably in the last half of this, which you're doing a lot now, looking at the brain. But anyway, yeah, so talk about some of your
Sure.
main findings there.
So, first of all, I'd say, the details of how mTOR has been activated by different types of amino acids or what amino acids those are, is, not really my work in any way,
Okay.
right? That was, very principally a lot of contributions were made by David Sabatini's laboratory at the Whitehead, looking at the detailed molecular mechanisms of how different amino acids activate mTOR. Um, and many other groups as well have contributed to some of those observations, both before and in the last few years. Overall, all amino acids probably activate mTOR, but they definitely have different strengths. And one of the most potent mTOR activators is an amino acid called leucine. Leucine is familiar to many people because it's one of the three branch chain amino acids. And so, something that was very big in the health field over the last 10 to 15 years, at least, maybe preceding that as well, was the idea that people should be taking dietary protein supplements either before or after exercise to enhance muscle growth and muscle hypertrophy and strength. And sometimes people were also selling supplements that were just the three branch chain amino acids, or maybe just leucine, or at least enriched in those amino acids and leucine. And so, and the rationale for doing so is that the branch chain amino acids, and particularly leucine, are very strong mTOR agonists. But that isn't my own work. It's just something that's been known for a long time, and many different people have contributed to.
Yeah.
Um, for the last 10 years or so, my lab has been looking at dietary protein and how it regulates healthy aging in a variety of different contexts. And the rationale for this started is something that many people had been working on for a while. So calorie restriction, where calories are restriction extends lifespan in rodents, was shown almost a century ago, more or less. Over a century, if you believe the British discovered it, less than a century if you believe that we discovered it in Cornell. But in any event, about a century. But actually, around the same time, maybe even a little bit sooner, it had been shown that protein restriction extends the lifespan of salmon. And after calorie restriction was discovered, people were interested in looking at, what components of a diet might regulate this. And dietary protein was found to have some of the same beneficial effects as calorie restriction. Didn't always work as reliably. And in hindsight, we can say that probably that's because some people were feeding the animals too little protein. And you certainly need a certain amount of protein and a certain amount of essential amino acids in the diet. And maybe both from a varieties of cutting that protein level too low, or maybe giving protein that wasn't enriched in all of the amino acids. Sometimes that didn't tend to work. But so people were really focused on calorie restriction for a number of years. My own interest in this came about from a couple of different ways. One is that Steve Simpson's laboratory in Australia did a very interesting nutritional geometry study that was published in 2014, led by someone in his lab, Samantha Solon-Beignet. And she had found that if you take mice and put them on 25 different diets, mixing all different ratios of protein to calories to fats to carbohydrates and looking at it in a very systematic way, the animals that lived the longest were ones that actually were eating a high protein, a low protein diet. And, that's not what we typically think of. We typically think of protein as something that's good for people. We advise people in this country to, generally speaking, eat more protein, particularly as you alluded to, elderly individuals, but, athletes and so on. Everybody says eat more protein. And that's because it builds healthy muscle. Muscle is good for resisting frailty and helping you live longer. It also promotes satiety. And so if you eat a very high protein diet, that's the basis of various fad diets like the Atkins diet, that you'll reduce your calorie intake and thus lose weight if you eat a high protein diet. And those things are all true, at least in reasonably short-term context. But epidemiological studies that have been done over the past 20 years or more say that actually the opposite is true, and that in the general population, adjusting for as many confounders as you think you can, generally speaking, people who eat more protein have higher rates of diabetes, as well as higher risks of cancer and potentially overall mortality as well. Um, and although this didn't inspire my work, a couple 2 years ago, a British twin study was published showing that the twin who ate more protein actually had a higher risk of sarcopenia, which is this age-related muscle loss. And so they actually grew frailer more quickly than the twin who was eating a low protein diet. And so, the question is like, how do you account for all of these possibilities? And so we had started working with Luigi Fontana. At the time, his laboratory was at Washington University in St. Louis, to sort of investigate, some of these effects. And what we found initially back in and published in 2016 was that both in humans and in mice, if you put humans on a protein restricted diet, where you took the level of proteins in the diet down for a human from about 17% to about 7 to 9%, so about 50% restriction, or about a 67% restriction in mice, which we used. Um, if you did that, you wound up with mice that were leaner, more glucose tolerant, had lower fasting blood glucose levels, sometimes had improved insulin sensitivity as well. And then using a series of amino acid defined diets, we were able to show that restricting branched chain amino acids alone could recapitulate some of these very beneficial effects. And so that's how we got into looking at dietary protein and the branched chain amino acids in particular. Now, it is also true that lowering branched chain amino acid levels or dietary protein also lowers mTOR activity in lots of tissues of mice.
And so that begs the question of what's the relative contribution of protein restriction to the, if you will, anti-aging, health span promoting effects of caloric restriction or intermittent fasting. And do you want to mention that, talk about the fact that probably most or all of the published studies on caloric restriction in rats and mice, the animals are actually fasting for an extended time period every day.
So the first thing I'd say is that calorie restriction and protein restriction are most likely quite different. And so there's a number of differences in terms of the phenotypes of what happens. But most notably, so calorie restriction generally involves restricting mice by 20 to 40% of their total daily calories. And so restricting protein by 20 to 40% really doesn't do very much.
Yeah.
And so John Speakman published a nice meta-analysis back in 2016 of many different calorie restriction studies and looking at them as a function of protein. And so what you can generally see is although there is a little bit of a trend, for the most part, the reduction in protein doesn't seem to be terribly driving the effects of calorie restriction. And I'd say that generally speaking, we think that's because there is sort of a break point. And so working in parallel with Jay Mitchell's lab, unfortunately, we were going to publish some of this back to back, but he died unexpectedly. Um, basically, what he and our group showed is that somewhere around about 9% protein is sort of where you activate the protein restriction effects, the effects of protein restriction. And so below about 9% protein for black 6 mice anyway, you get these effects where you switch over to activating different pathways and seeing the metabolic phenotypes of protein restriction. Whereas above that, changing that your dietary protein doesn't tend to have huge, very, very huge effects. And so one very interesting effect was that there's actually no significant differential gene expression in the livers of mice fed a 14% protein diet or a 21% protein diet. And so because all of those are sort of a protein sufficient state, it seems. Um,
But is there still a role for reduction in mTOR signaling in beneficial effects of daily time restricted eating or other intermittent fasting regimens? In other words, amino acids are one thing that activates mTOR. What about overall reduction in other nutrients or yeah.
So there is quite a convoluted set of issues around calorie restriction and mTOR signaling, and they relate to what you alluded to, this sort of fasting. So briefly, when we think about calorie restriction, some of the humans who started doing calorie restriction, some of them were eating 2 or 3 meals a day, but some of them were only eating 1 meal a day, and that's actually has something that's increased in popularity over time. But rodent studies of calorie restriction have almost always involved feeding animals once per day. And essentially, that's just for the experimentalist convenience. It's pretty labor intensive to feed the mice once per day. If you had to do that 2 or 3 times a day, it would be even more intensive. And you'd also be disturbing the animal's sleep because mice and rats like to sleep at night. Or rather, sorry, they like to sleep during the day, which is when, of course, people are around to feed them. And so typically, calorie restricted animals are fed once per day, most often in the morning, sometimes in the evening. Doesn't seem to make a huge difference in most studies what time of day they're fed. But the once per day feeding seems to play a really important role in some of the responses to things. And that's basically because the mice are hungry. They eat their food very quickly, somewhere between 1 and 3 hours, depending on the animal. And that means that they're fasting for probably 21 hours or so a day. And that's a pretty long time. It's probably more severe than a human fasting
Yeah.
for 24 hours, but that's something else that we don't completely understand. But in any event, this has two consequences. One is, at least in some of our studies, we found that this fasting period seems to be very important for the response to CR. And if we eliminate this fasting response using diets that are diluted in calorie content so they eat a lot more volume over a longer period of time, or where we feed them multiple meals, that prolonged fast is really required for both the full effect of calorie restriction on lifespan, the effects on frailty, the effects on molecular signatures in the liver and adipose tissue, and some of the metabolic effects. Not all of the metabolic effects. So calorie restricted mice with or without fasting are leaner, they weigh less, they have improved glucose tolerance. But that fasting is required for improvements in insulin sensitivity and these other lifespan and health span benefits. So the mTOR question is very complicated, though. So calorie-restricted animals become more insulin sensitive, which means that when they eat, they have a really high spike in mTOR activity. And so they actually spike much higher than an ad libitum fed animal that also eats a equivalent meal size. Conversely, at the end of their 21 hours or 23 hours of fasting, they've been fasting for a really long time. So they have lower mTOR signaling than mice that are ad libitum fed. And tracking these changes over time actually is something you can do, but all of the different experiments you might do have various different limitations. That said, my former graduate student Heidi Pack, who's now in a lab at UT Southwestern studying circadian rhythm signaling, actually found that at least in the liver, there's no overall change in mTOR signaling um, uh, it, resulting from calorie restriction, but there might be in other tissues like muscle. And so it could be that calorie restriction is certainly, it seems likely to regulate mTOR signaling. It certainly makes it more dynamic, but it also changes the pattern of mTOR signaling throughout the day. And it probably has different effects in different tissues.
And what about autophagy in under those conditions? Is it upregulated towards the end of the fasting period in, say 20 hours, 21 hours in mice?
I mean, that's a logical assumption to make. I don't remember for sure if that's what we found. Her paper was published, I think, last year in Cell Reports. So maybe we can look that up later. But I will say that calorie-restricted animals have a lot of interesting adaptations to the effects of the fasting.
Yeah.
For instance, and this is something that we haven't published yet, but one of my postdocs is currently writing up this manuscript. In calorie-restricted animals, you would think that there would be a big boost in ketogenesis because glucose levels and carbohydrates are less available, and the animals actually subsist on lipids for an incredible portion of a day. This is something that Matthew Bruss and others have shown a number of years ago. But turns out that mice adapt to that change. And so after a certain number of weeks, calorie-restricted animals no longer have upregulated ketogenesis during the day when they're fasting.
Okay, yeah, that's really interesting considering all the work being done on particularly beta hydroxybutyrate and its signaling functions and so on. Okay, so then uh, b- before we get to the brain, talk briefly about rapamycin and kind of the what's going on there. There's a lot of potential interest in translating the lifespan extending effects of rapamycin in mice to
Sure.
humans.
But I will say we didn't cover the other cool things about branched-chain amino acids, which
Oh,
is
okay.
that uh, in subsequent work after our initial work, we were able to show that branched amino acid restriction itself extends lifespan and reduces frailty in mice. And then more recently, we've sort of narrowed in on isoleucine as a key mediator of these effects. And so isoleucine restriction alone extends the lifespan of genetically heterogeneous mice by about 33% in males and about 7% in females. And in both of those cases, there's certainly a lot of concern that, because protein and branched amino acids are so important for muscles, that those animals would be more frail. They're actually less frail and don't have any significant defects in strength or grip strength or, rotor rod performance or other assays of that we've done to assess muscle function. And so it seems like, these could be really interesting potential interventions if we could figure out how they work, from a longevity standpoint.
So in the mice, you can, precisely control the amount of which amino acids and the amount because you're making up the diet. What are the implications for this for humans? So in other words, if I were to want to reduce the amount of isoleucine in my diet, what should I tend to avoid?
It's much easier to do a low protein diet. Vegan diet naturally has less isoleucine in it due to the fact that it has less calories from protein. But in terms of meats that contain less isoleucine, both turkey and emu are sort of the lowest of the major meats in
Hmm.
isoleucine content.
Emu. Have you eaten emu meat?
I've been trying to track it down. It is available here in Madison at the farmers market, but you
Yeah.
got to go at the right time.
Okay, that's interesting. Yeah, maybe I should try that sometime. All right. So then, all right.
Wanted
Let's
to do
see.
rat
You
mice.
also did some work on letting the mice, age and then giving the branched chain amino acids later in life. In other words, a lot of the early studies were more long-term, beginning when they're young, if I'm
So, in our when we're restricting all three branched amino acids, we show that starting very young worked, but that starting at 16 months of age, which is probably about 50 to 60 years old for a human, did not. In our more recent work with isoleucine restriction alone, we started at 6 months of age, which is about 30-year-old for a human, and that one worked brilliantly. In some work that we published a couple of years ago, we saw that a lot of age-related biomarkers still changed in 20-month-old mice with isoleucine restriction. So we don't know exactly what ages are most beneficial, but most dietary interventions in mice previously have been shown to work better in younger animals and be less effective in older animals.
So one of you've got, looking on PubMed, so you have several preprints up. In other words,
Mhm.
manuscripts you put up and then, presumably they're under review, or
Yep.
sometimes actually they leave both the preprint and the published thing up on PubMed. So you had one looking at giving animals a McDonald's diet, essentially. Well, I don't know if it's McDonald's, but Western diet, if you will, and then seeing if reducing protein can counteract the adverse health effects of a.
So, yeah, if you make an animal fat and obese by feeding it this sort of Americanized or Western diet, um, one of the things that's really cool is that you can reverse those effects by restricting either protein or branched chain amino acids or isoleucine alone or valine, which is another branched chain amino acid alone. Interestingly, leucine, which is the most potent mTOR agonist, restricting that doesn't do anything. So that's kind of an interesting thing that suggests that some of what we're looking at may be mTOR independent. But yeah, those effects are really dramatic and happen very quickly. And so the animals lose a lot of weight, particularly fat mass, and they have massive improvements in glucose tolerance and insulin sensitivity, even as they continue to eat more food. And so their calorie intake actually starts to go up when they are switched to these diets. And so eventually they're, essentially maintaining a lean phenotype, eating a very unhealthy diet where they're eating at least twice as many calories as mice that are still matched to them. They're eating a matched Western diet.
And that's high saturated fat plus simple sugar.
I think it's milk fat plus high sucrose plus some cholesterol for fun.
Yeah.
But we've also done at least a few of these experiments with a more traditional 60% high fat diet and seen similar effects.
Yeah, there's quite a bit of evidence that fructose in particular, maybe more than glucose itself, can have bad effects on metabolism and so on. It'd be interesting to see if, because we did some studies looking at the brain. I had one graduate student who did a lot of work on diabetes in the brain and looking at mechanisms and then looking at learning and memory and doing electrophysiology to look at synaptic plasticity. She also looked at neurogenesis. And so she found put fructose in the drinking water. They love that. And it does really bad things over a period of many, many months on their cognition and neuroplasticity. So it'd be
Yeah, one
interesting
of
to
my
look at.
students has been really interested in looking at dietary carbohydrates type and how that interacts with dietary protein. Um, Unfortunately, the results are too complicated to go into. But
Okay.
there are some interactions, but they're not simple to describe.
And also another preprint you have up, I don't know if it's published yet, was on cell senescence. I had Darren Baker on the podcast talking about senescence and focusing on the brain, but he's an expert on senescence in general. So you looked at,
Yes, so one of my new graduate students, Mariah Calbag, had recently shown, and we're working on getting that published, that branched chain amino acid restriction actually has some quite interesting effects on senescence, reducing senescence in the liver in a cell autonomous manner, but actually increasing senescence in white adipose tissue.
oh, well, that may be good. If, well, I don't know, because senescent cells are inflammatory and fats.
I don't know
Mm,
either. And
yeah,
then it's another interesting question about what senescence in white adipose tissue even means.
yeah.
But there's actually several people who have been publishing recently on white adipose tissue senescence as a thing.
And do you think in that model, when you're looking at the liver, that the sort of the senescent cells just stay there, or are they somehow removed?
That's an interesting question. I can't speak to that from our own research. I would say that, generally speaking, people think that senescence cell removal is important and that that's something that declines with age and that probably contributes to the effects. But I can't talk about that from our own research perspective.
Okay, let's go to the brain. You've done quite a bit. As you know, so back in the 90s, we done when I was still at the University of Kentucky, we did simple experiments, put rats or mice, depending on the model, on every other day food deprivation, so every other day fasting, and then let them go for a number of months. Initially, we used certain neurotoxins that selectively damage either neurons in the hippocampus by excitotoxicity, for example, or dopaminergic neurons with a mitochondrial complex 1 inhibitor, MPP plus, which comes from MPTP, and so on. And we found that intermittent fasting was very effective in protecting the neurons against stress. We also did a stroke model. Then when I moved to NIA, we did the same model you're using. Actually, we helped a little bit develop that model with Frank Lafurda, the 3X TGAD mice. And so these mice in our hands, they developed the amyloid and tau pathology and cognitive impairment fairly slowly compared to some other models. And so we started them on every other day fasting. We also did daily calorie restriction slash time restricted eating at 5 months, and then we let them go for a year. And then we found beneficial effects in ameliorating the cognitive impairment. And then you've been looking in the same model you have a number of studies, recent one last year in Nature Communications with protein restriction, but some other things. So talk about your work related to brain aging and Alzheimer's disease.
Sure. So, yeah, one of the things that we wanted to do was sort of explore how these might be interventions in Alzheimer's disease. Certainly, there's data going in to support that. About 10 or 12 years ago, Luigi Fontana and others had published some work showing that protein cycling was beneficial for the 3xTGE model. Which is sort of an intervention that is even less translatable than protein restriction, where basically every week or two the animals would switch to a zero protein diet for 1 or 2 weeks. And then they'd go back on a normal diet and then continue that cycle indefinitely. And so, we thought that this would be logically good for a number of reasons, including the fact that reduced mTOR signaling should be associated with beneficial effects just on healthy aging in general. And then that rapamycin, which is a drug that's an mTOR inhibitor, has been shown to have beneficial effects in the 3xTG model as well. And as well as maybe some effects on cognition in wild type mice. So we placed mice that are 3xTGs starting about 6 months of age, so after they've already developed some the pathologies of AD on either protein restricted diets or calorie restricted diets in the later studies or an ad libitum diet. And we showed that protein restriction had a number of beneficial effects. Basically, preserving cognition on a couple different assays that we did, reducing some of the Alzheimer's pathologies, including both A beta plaques and tau tangles, and doing some other things as well, including seems to have some beneficial effects on mTOR signaling, maybe autophagy, and inflammatory markers in the brain.
Yeah, so this is all very important. I mentioned at the beginning, it's not often scientists, go from studying some basic mechanism, so in yeast, to then all this pre-human work, I guess we would say, in the mouse models. And so are you involved interacting with people doing human studies? And how do you see your work and many other people working on this area?
So from a protein standpoint, there's been two published short-term clinical trials on protein restriction. One by Luigi Fontana that we helped in terms of data analysis and thinking about some of those results, and then a more recent one that was done on people with type 2 diabetes. And both of those showed independently that short-term protein restriction for something like 6 to 8 weeks resulted in weight loss primarily from fat mass, improvements in glucose tolerance, and particularly in the individuals with type 2 diabetes, improvement in insulin sensitivity. And so, that recapitulates very well the effects that we see in mice and seems to indicate that protein restriction could be beneficial for human health. Um, Now, interestingly enough, before insulin was invented slash isolated by, Banting and Best, there was a man named Kemp who actually put patients in very wealthy people anyway who had diabetes, and he brought them to maybe North Carolina, South Carolina, somewhere in the South, and put them on what he called a rice diet, which was about 2,000 calories a day, almost exclusively from rice and fruit. And, sure that it did not have nearly the success rate of insulin in terms of ameliorating their symptoms, but from what we can tell, it seemed to be beneficial for those patients. So there's a lot of decent, evidence, both recent RCTs as well as sort of as historical data that suggests that protein restriction can be metabolically beneficial. And so that is something that, I think is reasonable to think about going forward. There's a sort of a problem, one might think about, which is that, the most used clinically diet is that people are prescribed high protein when they're old and have sarcopenia.
Yeah.
And so there's a couple things to think about. One of which is that dietary needs and requirements may change over age. And in fact, the recommended dietary intake is different for males and females as well as people of different ages. There's also the fact that many people who are elderly are actually protein deficient. And so when we think about protein deficiency, that's different from protein restriction, right? So protein restriction works, generally speaking, by taking people who are eating more protein that they need, or animals are eating more protein that they need, and lowering that.
Hmm.
In a protein deficient state, someone is eating so little protein that they are not getting enough of the essential amino acids that they need, which means that their body has to break down skeletal muscle in order to liberate essential amino acids, because that's the only source of amino acids in the body, or the major one. And protein deficiency is something that is suffered by a number of elderly people for a couple reasons. One is that protein is expensive, and many people who are older are eating on a limited budget.
Hmm.
And so they may choose foods that are higher in carbohydrates because they have more calories. A second is that Many older people have what's called anorexia of aging. Basically, they're not as hungry. And so, certainly I've noticed that in my own grandparents' lives that they became much less hungry as they grew older. I think that's sort of a general common experience for the elderly. And so, if you're eating less food and you're eating food that tends to be higher in carbohydrates, you may be eating very little protein. And in that perspective, you may need to be prescribed a higher protein diet just to bring you up to protein sufficiency. So you're not wasting down skeletal muscle to make up for a nutritional problem. So
But
those are some of the
so
complications.
Dudley, so what about uh it's important to exercise too, right? I mean, you can have a high protein diet, but if you're not exercising your muscles, uh they're it's going to be hard to maintain their mass.
Well, that's a very interesting question. And so we're exploring this in a couple different age groups of mice now. But, one of the big paradoxes that we had is that the humans who particularly eat a high protein diet, the athletes and the bodybuilders, right? We know that even though they're eating a very high quantity of protein, we know that they don't have a very high rate of diabetes. And so the question that we want to address there is why. And so the simple explanation that we had for a long time was that exercise probably protects against the metabolic effects of protein. Looking at that in an animal is actually quite tricky because the easiest way to make an animal exercise is to put them on a running wheel or a treadmill. And animals that run a lot are lean. And that's sort of hopelessly confounding for our purposes. Fortunately, working with people here at Wisconsin, we were able to first help develop a model that was made by Troy Hornberger here at Wisconsin, and then use that mouse model of resistance exercise training to look at what happens to animals as they undergo resistance exercise. And there are some interesting things, and I'll say, animals put on a high protein diet that are sedentary tend to get fat, and we see that very dramatically. If they're doing resistance exercise training, they tend to stay leaner. They also build big muscles if they're doing resistance exercise training. They gain strength more quickly than animals on a low protein diet, even if those animals on a low protein diet are exercising. But I will say at the end of the day, if you take them out until their strength gains plateau, the low protein animals catch up. And so low protein fed mice that are exercising become just as strong as high protein fed animals that are exercising. They don't get big muscles. So people who want big muscles just for aesthetic reasons or so on probably do benefit from the high protein diets in terms of promoting muscle hypertrophy.
Hmm.
But in terms of muscle strength, the people on the low protein or the mice on the low protein diet that are exercising catch up quite quickly with the high protein fed individuals.
There's some work in humans too, Antonio Paoli and others that there's actually quite a few studies now looking at whether intermittent fasting has any effect or uh, impairs or enhances the effects of resistance training. And kind of the take-home message, if you put all what's out there together, is it definitely doesn't have any adverse effect as long as you're exercising. It kind of makes sense from all this kind of makes sense from an evolutionary perspective, right? I mean, we and other animals evolved in environments where food was generally scarce and we had to work for it. And so eating patterns were more sporadic. So our systems are kind of adapted
Definitely.
to intermittent eating patterns. And even they think about predators, right? They may go like in the winter, right? In Wisconsin, so deer are a prey animal for wolves, right?
Well, I mean, there's definitely deer walking around in the winter, but I certainly imagine that it's more difficult to find prey in the winter.
Yeah, and the wolves can go for a week, 2 weeks without killing a prey animal, right? So they're fasting for a week or so, and they're fine. But anyway, so uh, all right, but so one thing then, I think you and I both agree that we're not going to spend a lot of money on buying protein supplements.
I think that's probably true unless you have some particular need.
Right. And yeah, this is a big thing. There's an untold number of companies, right, are selling protein. Um, and so what about, so you've got, this is always a problem. It's a big problem with our healthcare system in general, right? You've got all these profit-driven forces, if you will, encouraging people to, either eat bad food by by advertising it, then making it not too expensive, making it highly palatable, or, advertising the protein, saying you're going to, be strong and healthy. Um, Education is the way to, one way to combat that, but it's kind of hard, right? So in other words, how, I'm doing this podcast, which, I'm not Joe Rogan, so we don't get millions of viewers, and the people that are viewing this podcast are really interested in science and learning science. Have you thought about this? How do you go about changing, when it gets to the point that the science says high protein is not good for health? Maybe you don't think we're at that point yet, but say we are. Then what happens then? How does it make change?
Well, I just came back from a nutritional meeting where there were many, many people who were talking about the health benefits of protein and different types of protein in terms of plant versus animal and so on. So definitely, there's plenty of people who are, can point to great examples in the literature of, protein being beneficial. And I think there's no doubt that at least in short-term studies,
Yeah.
protein and individual amino acids, when taken along with exercise, help promote strength gain and muscle mass gain and recovery. So I think that, there's definitely a role for protein and, it is essential in the diet, right? There's 9 essential amino acids in the human diet. And if you don't have enough of any of them, very bad things will happen to you very quickly.
Yeah.
So, protein is very important. I think it's a, open question as to exactly what type of incentives need to be, put in place from a public policy standpoint to promote healthier foods rather than unhealthy foods.
Yeah.
Fortunately, unhealthy foods tend to be very delicious. So that is an issue. And I think that take a lot of thought to think about this. Of course, it's also very interesting, right, that things like GLP-1s are, changing not just
Yeah.
how much people eat, but apparently the composition of their grocery basket as well, because apparently people on GLP-1s prefer foods that are, lower in sugar and might actually be healthier. And so now I was reading that, there's interest in promoting smaller amounts of more expensive foods to those individuals. So that if you're only going to eat a little bit of steak, maybe you should be eating filet mignon now as opposed to, hamburger.
Yeah, those are the GLP-1 peptides are amazing. Seems like what they're doing at people with overweight, diabetes, good for cardiovascular. It's strange. My background is started out in endocrinology, then neuroendocrinology, and all hormones are, they're released, they're circadian effects, and then they're released in response to some stimulus, oftentimes, stress hormones in response to stress, and GLP-1 in response to food intake, particularly carbohydrates, sugar. And so normally you eat a meal, the cells in your uh, GI system release GLP-1 into the blood. It stays in the blood actually a short time period and it's removed. And evolutionarily, you'd think there must be a reason for that. And so I'm always concerned with interventions uh, like where you get like a constant level, constantly activating a receptor. So we'll see. Maybe there won't be any bad side effects. So far, it just seems to be nausea and some other things. Now, you've done some work on, I think, right, intermittent dosing. So you said early on that it's essentially it's generally not good to have mTOR constantly activated throughout your life. You know, intermittently is better. And you've done that's something we've thought about a lot too, is some intervention that we thought about it from inducing like a metabolic stress. So we did a lot of work with 2-deoxyglucose, for example, founding give it once a day to animals. It does good things for their brain. And when you give it once a day, you get it's kind of like transient daily calorie restriction, the time course of it. But then we put it in their food, and when the animals were fed ad libitum, and long term it was bad for them. Can you talk about the idea of with certain interventions, and this is very different thinking than the pharmaceutical companies think about intermittent dosing, because I think it's could be something very important in the future.
Well, I mean, that's a good enough reason to change over rapamycin that you had wanted to talk about. Um, and
Yeah.
so, rapamycin, I think probably many of your viewers are familiar with it. Generally, it's an mTOR inhibitor that was originally discovered on Easter Island, or as the natives call it, Rapa Nui. And it's currently available in two different commercial formulations called sirolimus, which is just rapamycin, or a modified version that's called everolimus. And so, rapamycin treatment of mice has been shown since I think it was in 2009 that Dave Harrison and the ITP showed the extended lifespan in mice. That's subsequently been reproduced by probably 10 or more labs across the globe, and in many different varieties of mouse strains and backgrounds. It also has beneficial effects on all sorts of different mouse models of diseases and aging, particularly some cancer models, but other types of models as well. Um, and so one of the things that's quite interesting about rapamycin is that it's primarily an inhibitor of mTOR complex 1, which is what most people think about as mTOR and is also the amino acid sensitive complex. But in chronic dosing, it also inhibits mTOR complex 2, which is a second mTOR complex. And the reason for that relates to both the structure of mTOR complex 1 and mTOR complex 2, as well as the pharmacokinetics of rapamycin. But essentially, if rapamycin hangs around long enough, it sequesters newly synthesized mTOR and prevents the formation of new mTOR complex 2 as that endogenous complex degrades. Even though rapamycin is not capable of binding directly to a ready assembled mTOR complex 2. In contrast, mTOR complex 1, it can bind directly to essentially instantaneously and inhibit mTOR very quickly. And so, the time courses sort of vary for this, but generally speaking, after a couple days in cell culture or probably something like 3 days to a week in a mouse, you inhibit not only mTOR complex 1, but if you're giving new rapamycin every day, you're also blocking mTOR complex 2 activity. And while mTOR complex 1 inhibition alone is capable of extending lifespan, and that's been shown in genetic models using flies and worms and mice, inhibition of mTOR2 genetically is almost exclusively bad. And so a lot of that work is work from my lab, where we knocked out key components of mTOR complex 2 in either the whole body in an inducible complex, or we deleted one copy of that in the whole body, or we deleted mTOR complex 2 in either the liver or the adipose tissue or the hypothalamus. Generally speaking, it seems that mTOR complex 2 inhibition leads to metabolic dysfunction, particularly glucose intolerance, but sometimes insulin resistance as well. And also leads to, depending on exactly where you're inhibiting mTOR and how, either increased frailty or obesity or both, and shorter lifespan, particularly for males. And that lifespan deficit can be as 40% or more.
Wow.
So it can be a pretty significant effect on longevity. And in humans, rapamycin treatment is associated with a number of different side effects. And these side effects are all dose and time dependent. So the more rapamycin you take and the longer you take it for, the more likely you are to exhibit these effects. And I should say the rapamycin is primarily prescribed as an immunosuppressant, which is often dosed as something like 1 milligram per day, but can be higher. But sometimes has been dosed in cancer trials much higher, and it's also dosed to people with tuberous sclerosis who have a genetic disorder where they have hyperactive mTOR signaling all the time at much higher levels, somewhere 5 to 10 milligrams per day or more. Um, and so some of the things that rapamycin does in terms of side effects, so first of all, it's an immunosuppressant, it's prescribed for that. But also and so any immunosuppressant is going to raise the risk of various different infections. And so in humans, it does increase the risk of bacterial and fungal infections. Some people who are taking rapamycin for a long time get these, and these can be quite life-threatening, and can include pneumonia and sepsis. And a number of individuals over the years have died from when they're taking very high doses of rapamycin, particularly for things like tuberous sclerosis, but also in cancer trials. It's always difficult when you're giving rapamycin to someone who is already ill to determine the cause of death. So, but it's estimated that somewhere between 2 to 5% of people who are taking very high doses of rapamycin may have died in some of these trials. On the other hand, rapamycin at lower doses, an immunosuppressive regimen, has often been given to many people for years safely. Rapamycin also is associated with higher risks of new onset diabetes, glucose intolerance, and hyperlipidemia, so high blood lipid levels. We don't really know what the long-term consequences of those are, except that, diabetes is obviously bad. But almost all immunosuppressants are associated with an increased risk of new onset diabetes, so that's not exclusive to rapamycin. And the high levels of lipids would probably be bad, and we would probably say that that relates to cardiovascular disease. But most people who are clinically being followed with rapamycin, particularly for immunosuppression, if they develop hyperlipidemia, they'll be simply switched to a different immunosuppressant, and then that effect will almost always, probably always, I think, go away. Um, these effects can be observed in mice as well. Mice have impaired glucose tolerance, impaired islet function, and high blood lipid levels if you give them enough rapamycin for long enough. So these are all things that people are concerned about. We think that most of those effects involve the inhibition of that are bad, involve the inhibition of mTOR complex 2. So mTOR complex 2 is certainly contributes to the immunosuppressive effects of rapamycin, and we know the biology behind some of that. We know the biology behind some of its effects on diabetes. My lab had shown or rather, I'd shown when I was in David Sabatini's lab that leads to hepatic insulin resistance. We've also shown in my own lab's work that inhibition of mTOR2 in various tissues is associated with high blood lipid levels. All of these side effects seem to be associated with mTOR2 inhibition. So going back to what you asked about intermittent treatment, we had theorized that if you gave rapamycin less frequently, you gave essentially a shot of rapamycin, it would circulate through your blood, it would inhibit mTOR complex 1. That inhibition is very stable. Once rapamycin and mTOR bind together, essentially they don't come apart at all. The off rate is very slow. And then the rapamycin that wasn't binding to mTOR would just be excreted over the course of, a few hours to days. And then mTOR2 would probably not be affected very greatly because by the time that mTOR2 is ready to be inhibited, there wouldn't be a rapamycin that was free floating around the blood anymore. And roughly speaking, that seems to be true in mice. So if you give mice rapamycin less frequently, those side effects become less severe or absent. And so we showed that if you give rapamycin once every 5 days to a mouse, it becomes glucose intolerant. It still has effects on the immune system, but they're not as strong. And that the effects on blood lipid levels go away. And so we were able to show that that same dosing regimen still extends the lifespan of mice. And so many people, there's about, as far as we can determine, probably 3,000 people or so around the United States who are taking rapamycin off-label for geroprotective effects because they believe it will help them. Um, Alan Green, who recently passed away, a doctor in New York City, was treating about 1,500 of them. And overall, most of those individuals were taking rapamycin intermittently. So taking rapamycin or maybe sometimes everolimus um, either once a week or once every 2 weeks. And so typically they would take a relatively higher dose, something like 5 milligrams, I think, in a survey done by Matt Kaberlin, something like 5 to 6 milligrams once a week, or maybe 5 to 6 milligrams twice a week, or once every 2 weeks, or maybe even higher doses once every 2 weeks. But the same idea that you would hit mTOR complex 1 really hard, and then you would wait, and those beneficial effects might continue for some amount of time, but you wouldn't have these off-target effects of mTOR complex 2 inhibition.
But this is an actual, they're not a part of any study, so do they have baseline data and data at different time points after starting intermittent rapamycin?
Well, it's kind of an interesting population,
Yeah,
and I don't think anyone's done a formal demographic analysis, but
yeah,
you can imagine that this population is very interested in monitoring its own health, and so
yeah.
its health is
It's
probably fairly on the good side of things to start with.
yeah,
Many individuals are eating either low-calorie diets or
yeah,
ketogenic diets or other diets that are healthy
yeah,
and that they're socioeconomically better well off.
yeah.
Buying rapamycin off label and paying a doctor cash to, prescribe rapamycin is not the most expensive thing in the world, but certainly not within the reach of everyone. And so my lab has been running an observational study now of some of these individuals, and hopefully we'll be able to publish on that within the next year or so. And generally we'd like to be able to follow some of these people long term. Um,
What you do, you have to get them to go off, get half of them to quit taking rapamycin, the other half keep taking it. And
it turns out from an IRB perspective, that is that changes things pretty dramatically.
yeah,
Because an observational study of individuals who are choosing to self-administer a drug is low risk and sort of largely, but not lar in the lowest risk category of an IRB study.
yeah.
Where if you are prescribing someone an immunosuppressant and then following them that's a much more serious issue.
Yeah.
So with Adam Konopka's lab at UW-Madison, we're actually conducting two randomized clinical trials of everolimus and rapamycin for different purposes. There's a number of groups around the country who are also conducting short-term RCTs of rapamycin. Every one of those involves site visits and safety monitoring where all the individuals are being continuously assessed for a variety of safety panels
Yeah.
and self-reporting side effects. And then those side effects have to be referred to an independent physician who might decide to unblind those individuals, might decide to take them off or put them on treatments or so on. So that's a much more serious issue than an observational study. For our observational study, what we have done is we have recruited some individuals who are already planning to take rapamycin but have not started yet.
Yeah, that's good. What's going on with the dog aging rapamycin project? I haven't been following that at all.
Well, the dog aging project itself, as I understand it, lost NIH funding for a couple
Uh,
of years. The rapamycin arm of that study, I believe, continued with private funding, and I think it recently got a new R01 award that Roz Anderson at UW-Madison is one of the PIs on. I think the lead organizations is in Texas, and I think most of the vets are probably based out of Seattle.
well, let's hope there's some interest in aging. There should be by the current administration. I think funding's going to obviously be shifting around, and I don't know, but everybody's interested in aging. Nobody wants to get old and die, so I think that there's a lot of opportunity for good grant proposals.
Could be, I think, definitely there's a lot of uncertainty right now. And, um, I think the
Yeah, and
thing,
it's I know so I've been talking to Rafa DeCabo and other colleagues at NIA, and they've already lost people, tenure track investigators, so the intramural program's definitely already affected, and the indirect cost thing that's going to affect if that if that happens, it looks like maybe it will. I don't know. So we'll see.
I mean, uh, now we're supposed to, since indirect costs are a little bit mysterious, it's recommended that we refer to them as facilities and administrative costs, to make that more accessible, to the general public.
Yeah,
But I think, that sort of long term at least seems to be because that's frozen by the courts at the moment.
yeah,
I think the fact that NIH funding overall seems to be slow coming out and the issues with study sections and councils are, something that most people are not like thinking about, but have really serious consequences. I'll say if just one of the consequences is one of the graduate programs I'm affiliated with just decided that instead of admitting about 30 students, expecting
wow,
probably about 20 to show up,
yeah.
they're not going to accept anyone who hasn't already been taken into a lab. So essentially, they're doing direct admits this year.
That's happening in a lot of
And
places.
that's happening
Hopkins
nationwide.
hasn't done it, but they're thinking about it. And this the thing is that this could be transient. It's just unclear. And I hope that we don't want to discourage people, young people, from going into science. That'd be a terrible thing, right?
Well, I mean, I think the biggest issue is the uncertainty. Nobody can plan when we don't have good data on what's coming.
Yeah.
And I mean, in my own case, I thought that everything was going to be fine financially, and either things will be fine financially in the fall, or my lab will be hundreds of thousands of dollars in the hole. And I can't tell which because there's, that's relying on decisions that haven't been made yet.
Yeah. Oh boy. All right. So I guess we can finish there, or maybe we should talk for 2 more minutes on a high note.
Sure.
What are you doing now on are you moving towards doing a lot more things relevant to the brain and aging?
I mean, right now we're working on continuing and finishing our analysis of some of the effects of branched amino acid restriction on Alzheimer's disease, and that's something that we've been working on for some time. The results are quite interesting, in that all three of the amino acids definitely have effects on different aspects of pathologies of Alzheimer's disease. It's not easy to summarize what exactly the overall effects are, but that's something we're still looking at. And we have a preprint out showing that it seems that fasting is probably also important for the effects of calorie restriction on the brain and cognitive health as well in mouse models of Alzheimer's disease.
I
So
remember
that's
way
definitely
back when
something
they
that we're
were
very interested in.
I remember way back when people were just getting started looking at, essentially transcriptomics, looking at all the messenger RNA levels of all genes in cells. And Tom Prola there, uh, I think, published one of the first studies looking at gene expression in the brain during aging. Are you trying to get at, what's going on in terms of gene expression, protein levels, to try to get more mechanisms beyond this, say, an effect on mTOR? What else is going on? And of course, the brain is complicated. There's neurons, glia, and so on. So it seems like there's a lot of opportunity to of endpoints, a lot of different endpoints in the brain tissue from these animals that you're putting on
I
low
mean,
brain.
I think there would be an enormous amount of interesting things that one could do in terms of longitudinal studies and different tissues, including the brain, single cell studies, and so on, to try and understand, exactly what's happening when we modify just, these simple dietary components and see these dramatic effects on health span and longevity. And, I think it'd be great to, be able to do more of that. Obviously, that's funding dependent as
Yeah,
well.
yeah,
So it'll be
yeah.
interesting to see, how funding availability changes over time, particularly for sort of this health span and aging research.
Well, I hope you get funded. Your work's, in my view, like really right at the cutting edge, and in this area with a lot of implications for you, me, everybody in terms of their health as they get older. So thanks a
Thank you
lot.
for those kind words. And yes, I mean, hopefully, uh, aging and health span research and the kind of work that we and others are doing, will be continue to be appreciated.
Yeah. Well, it certainly is by the people that know about it. So thanks again, Dudley.
Thank you. Take care.
Yeah. Bye.
Bye.